Evnr 3200, Presentation 13, Meiosis and Alternation of Generations. Even though the title of this particular presentation starts off with meiosis, which is the word that's figured in the title of the textbook chapter, we are going to be focusing primarily on the alternation of generations life cycle that's characteristic of plants. And we will deviate quite a bit from the chapter in the book, which is pretty brief. So I'm just letting you know we're not going to go into meiosis in detail. We're not going to learn the phases of meiosis. You did that at some point in your dim, distant past. I'm not worried about that. I am more worried about you understanding the details of the sexual life cycle characteristic of plants, and we will look at that particular life cycle as it's demonstrated in the mosses, as it's demonstrated in the ferns, as it's demonstrated in the pines, and as it's demonstrated in the flowering plants, using the lily as an example. And once we're done with that, it's going to make life much easier when we start taking a look at some of the plant diversity presentations that we'll be looking at later on in the semester. So understand we are focusing a tremendous amount in this particular presentation on the alternation of generations life cycle, and it is going to depart significantly from what is found in the book. But standard operating procedure, you're going to wind up focusing on the content in this presentation anyway. And I will strongly suggest that if at any point you're not necessarily keen on the illustrations I've chosen, or you're not understanding what the content's all about, please Google any or all of these life cycles. What you will find online is not appreciably different from what I am giving you. Yes, there might be a little bit more detail, but by and large, it's pretty much the same thing. And everything you get online will be accurate, and everything you get online will meet my expectations for what's found in this particular presentation, and should meet my expectations as far as what's going to be on an exam. So let's start off and talk about sexual life cycle basics. The far majority of organisms that are qualified as eukaryotes. Now remember, a eukaryotic organism is one that has nuclei, one that has membrane-bound organelles in its cells, and the cells are larger and they're more complex, and these would include things like plants and animals and fungi and protists. Well, most of those eukaryotic organisms feature sexual life cycles. And a sexual life cycle is one where the mature organism, more often than not, is diploid. Animals are diploid. Plants are diploid. There are some exceptions. However, we're taking a look at our slide here. Plants are diploid organisms. That means your typical plant has two sets of chromosomes. That means they have two sets of genes. That means the genes that are found in that plant is going to be manifested as a wide variety of possible characteristics. And those characteristics the plant has fits it to its environment and improves its ability to survive. All right, so where do you get those two sets of chromosomes? You get them from an event of fertilization. So therefore, a plant is a product of fertilization. All right, so let's take a look at our own life cycle. I know we're supposed to be talking about plants, but maybe the best way to start is to take a look at the typical animal life cycle, which is indicative of human beings. The sexual life cycle of human beings begins with adults. I see down there in lower left. And I know our adults feature gonads, ovaries, and testes. Our adults are separately females and males. And in those gonads, the ovaries, and the testes, an event called meiosis takes place. You probably learned about meiosis once upon a time. It's called sexual cell division, or sometimes it's called the reduction division. In any event, what it does is it takes the diploid cells in the ovary or in the testes and generates haploid products. In the case of the ovary, you're going to make a haploid egg. In the case of the testes, you're going to make haploid sperm. Now, as far as humans go, we've got a grand total of 46 chromosomes arranged in 23 pairs, which means an egg is going to have one set of 23 chromosomes, and a sperm is going to have one set of 23 chromosomes. And as it goes, the sperm fertilizes the egg cell, a process known as fertilization, or more technically is referred to as syngamy, Syngamy, and we might have explained that once upon a time. Syn, the prefix, means together. Gamete refers to the word gametes, Gametes, which refers to sex cells. Sperm and egg are gametes. So an event of fertilization or an event of syngamy takes place. A sperm cell containing 23 chromosomes fuses with an ovum, an egg cell, containing 23 chromosomes, and you get a brand spanking new diploid cell called a zygote, featuring the grand total of 46 chromosomes, and that zygote will divide by mitosis, routine, general, somatic cell divisions, and will create our adults all over again. So any sexual life cycle, and it doesn't matter whether it's the animal cycle that we're looking at here, as exemplified by humans, or plant life cycles, or any sexual life cycle, there's always two critical events. And those two critical events are meiosis and syngamy. Syngamy, i.e. fertilization, and meiosis, i.e. sexual cell division or the reduction division, where we actually take something that's diploid and create haploid products. Now what we're going to do is we're going to take a look at more detail in terms of this process of sexual cell division in plants. Now there's one more little bit on this slide I want to talk about, and that's where it says meiosis results generally in the formation of sperm and eggs in animals. We know that already. But when we think about it in plants, a diploid plant, like a pine tree, or a maple tree, or a lily plant, is going to make pollen and ovules. Pollen constitutes the male part of the sexual life cycle, and ovules constitute the female part of the plant life cycle. You can think of pollen as being tiny haploid male individuals, and you can think of ovules as being tiny haploid female individuals. Now, if you're haploid and you're being produced by a diploid plant, you have to be produced by an event of meiosis. So the parallel here is in animals, we make sperm and eggs by meiosis, but in plants, we make pollen and ovules by meiosis. And you hold that thought because we're going to be dealing with it when we start talking about the alternation of generations life cycle. On this slide and the next, I'm going to feature some schematic diagrams going to help us understand the structure and the operation of the sexual life cycle in first, animals, as exemplified by humans, and second, in plants. Now let's take a look at the life cycle found in animals. It's commonly referred to as the gametic meiosis life cycle. Now that's not a different language, but what it really means is it means that in this life cycle, gametes are made by meiosis. So the adult at 12 o'clock is diploid. Diploid in my diagram here is colored green, and the adult makes sex cells through a process of meiosis. Those sex cells are called gametes. They're made in sex organs called gonads, and I notice that my color has changed to kind of a peach color, which is indicating haploid to me. I've got an egg, I've got a sperm, each represented by one N, meaning one set of chromosomes, and they undergo syngamy, i.e. fertilization. I get a zygote, the fertilized egg, which grows by mitosis into a brand new diploid adult. So I noticed that this sexual life cycle is characterized by those two events I talked about previously: syngamy, or fertilization, and meiosis. And in this case, the meiosis results in the formation of egg and sperm. Now on our next slide, we're going to take a look at the sporic meiosis life cycle demonstrated by plants. And we'll understand the sporic meiosis life cycle goes by another name. And it's going to look a little bit similar to this, but it's going to demonstrate some very big, significant differences that make all the world of difference to a plant. Okay, well, let's look at this sporic meiosis life cycle. The sporic meiosis life cycle is given that name because spores are produced by meiosis. As a matter of fact, if you look at the life cycle diagram, you'll see meiosis over there on the left, and you'll see it does produce something called a spore. That's why we call this sporic meiosis. But the other thing is this life cycle is more appropriately referred to as the alternation of generations life cycle, and it is indicative of plants. So you will see alternation of generations as the very last bullet of my list of points there. And the reason why we call it an alternation of generations life cycle is there's actually two individuals in this life cycle. There's a haploid individual called the gametophyte, and you see it up there at 12 o'clock on the life cycle. And there is a sporophyte or a diploid individual that I see down there at 6 o'clock on the life cycle. And if you follow the life cycle, we alternate between the haploid generation, the gametophyte, and the diploid generation, the sporophyte. Now, those generations, they get their names in terms of what they make. The gametophyte literally means gamete-producing plant. The suffix phyte, phyte, means plant. Well, if I'm looking at my life cycle, I notice the gametophyte, which is haploid, makes eggs and sperm, which are also haploid. And those eggs and sperm are going to unite through syngamy to create the diploid phase of the life cycle that I call the sporophyte. And the sporophyte gets its name because it is a spore-producing plant. And therefore the spore is produced through the event of meiosis. Well, hold on, Dr. Knauss. I got one question here. You mentioned on the previous slide that in plants, meiosis create pollen and ovules. All right, well, what we need to do is we need to start in a very general sense, because pollen and ovules are only a feature of seed plants, which would be pine trees and lilies. So that's something let's put it this way, that is a bridge that we will cross later on. At this point, make sure you understand that a sporic meiosis life cycle is also known as an alternation of generations life cycle, is an alternation between a sporophyte generation that makes spores by meiosis and a gametophyte generation that makes gametes. And the gametes undergo fertilization and create the sporophyte, and the sporophyte generates the spores by meiosis, gets me back to the gametophyte generation. So I'm going to run down the bullet points just to make sure that we got it down here. There are two phases, one called the sporophyte, which is diploid, and one called the gametophyte, which is haploid. The sporophyte makes spores by meiosis. The sporophyte is illustrated at 6 o'clock. The process of meiosis yields spores. I see there at 9 o'clock. The spores develop into the haploid gametophyte. The gametophyte makes gametes by mitosis. Now, the reason why the gametophyte makes gametes by mitosis is the gametophyte's haploid, and the gametes are haploid. So you can't have one set of chromosomes. You have to basically create two brand spanking new sex cells that have the same set of chromosomes that you had in the gametophyte. The gametophyte makes gametes by mitosis. Syngamy of those gametes results in the formation of zygote. Remember, that's a brand spanking new diploid cell, and a zygote grows into a new diploid sporophyte, alternating between these two generations. This is the life cycle that is typical of plants. And I strongly suggest that this becomes an indelible part of your memory, because it is going to have application over and over and over again, not only in this presentation, But in future presentations, when we start taking a look at mosses and ferns and cone-bearing plants and flowering plants, it might even have applications when we start talking about fungi or maybe some other organisms. So make sure you have full command of this. And believe me, we will visit it over again and again and again on the next bunch of slides as we go through this presentation. So let's take a look at the life cycle of one of the more primitive plants, the moss. Moss plants, you might remember, are real small. They have no vascular tissue. There is no xylem, there is no phloem, there is no cuticle. And we're going to find a few more interesting things about the mosses. Now, before we even get into the life cycle, and we're going to cover it over a series of slides, we're going to take our time with it. Please keep in mind that the moss life cycle is going to follow the sporic meiosis or alternation of generations model. We're going to see a gametophyte and we're going to see a sporophyte. We will see an event of meiosis leading to the formation of spores, and we will see an event of syngamy or fertilization leading to the formation of a zygote or brand new diploid individual. The same hallmark features of the alternation of generations life cycle we just looked at a moment ago will be seen in this life cycle. So here I've got a picture of a moss, bottom left, and that's a male moss. Mosses come in two sexes, males and females. The mosses that we actually see in nature are haploid, so technically they're gametophytes. They're small, they're non-vascular, they don't have cuticle, and they live in very moist habitats. Now, at the top of the male moss, we're going to find special gametangia, or gamete containers, that are called antheridia. A gametangium is a gamete container, and the official term for a male gamete container is an antheridium, antheridia plural, and an antheridium will contain haploid sperm. Now, important point to be made, and you might want to note this next to that bullet point on this slide. Those haploid sperm are made by mitosis because it's a haploid individual making haploid cells. You cannot perform an event of meiosis here. So even though meiosis is a process that creates haploid sperm in humans and in other diploid organisms, in this case we've got a haploid male gametophyte making haploid male gametes, sperm, and the process needs to be mitosis. So walk me through the pictures, Knaus. Well, the male moss plant's on the left, and that illustration right next to it is a microscopic view of the top of the male moss plant, and a little blue arrow is indicating the presence of a sac-like structure, which is an antheridium, and in that antheridium I am going to find sperm cells. On the far right, I see a picture of the sperm cells found in mosses. They're very unique. They've got multiple flagella, and they're kind of curly-cued. That is technically what a moss sperm looks like, and it will be involved in an event of syngamy. But our next slide is going to deal with the way the females operate. Once again, the moss plants that we see in nature are haploid. We would call them gametophytes, which means gamete-producing plants. There are male moss plants that make sperm. There are female moss plants that make eggs. So our moss plant photographed below left is female moss plant. And if I take a look at the top of the female moss plant, I am going to find female gamete containers. Female mosses make female gametangia. Now remember, a gametangium is a gamete container. Females make eggs. So the female gametangium gets a special name. It's called the archegonium, or archegonia plural. And it looks like a bud vase, like something you'd put a rose in. And at the very bottom of the structure, I see a little chamber. In that little chamber is an egg cell. So I'm looking at the microscope picture in the middle. I see the blue arrow indicating an archegonium, and it does look like a long-necked structure with a chamber in the bottom, and you see a little dot, which is the egg cell. Then I've got an artist's rendition of the same thing over on the far right. So we're setting up a situation where we're going to have an event of fertilization or an event of syngamy. Somehow we're going to get a sperm cell from the male to the archegonium of the female. That sperm cell is going to travel down the neck of that archegonium and fertilize the egg down in the bottom. Let's see what happens next. Once again, I will remind you that moss gametophytes are small, they're non-vascular, they don't have a cuticle, and necessarily they are going to live in very moist habitats. Obviously, some mosses dry out sometimes, but for the most part, they live in habitats where there is some amount of moisture. And that moisture will occasionally form a complete film over the surface of the plant. When the mosses are covered with a layer of moisture, sperm are released from the antheridia on the males. And those sperm will swim their way through that film layer of water to the archegonium of the female. The egg will be fertilized and will become a zygote. Now keep in mind that zygote will remain in the base of the archegonium, and it will develop in the base of the archegonium. So taking a look at the pendant drawing over on the far right hand side, you will see the archegonium with its egg. You'll see the sperm approaching the egg. You'll see the fertilized zygote in the base archegonium, and that will divide and start to grow into a diploid individual, which will be positioned, located, and situated on top of the female plant. So, since this slide deals with the issue of syngamy, you might want to write down the word syngamy just to send that particular term home concerning the content on this particular slide. This is fertilization. This is syngamy. All right, now after the event of syngamy or fertilization, things get a little different. The embryo develops at the top of the female plant. As I said, it is located in the archegonium. It has not gone anywhere. But this diploid plant is a sporophyte. It's a brand new generation. Now, take a look at the illustration immediately below that bullet point on the left of the slide. You'll see the gametophyte, the little green leafy-ish gametophyte. But then you'll see this long, skinny stalk with a capsule at the top. And that thing is the sporophyte. That's the diploid generation. It has grown from the fertilized egg, became an embryo, developed into a mature sporophyte that you see right there that is nowhere near the same in appearance as the female. The sporophyte is totally dependent on the female. The sporophyte consists of a really long, thin stalk called a seta, and on top we'll find a capsule. The capsule is more officially known as a sporangium, or a spore container, and that spore container is ultimately contains spores. Now, what I know about the alternation of generations life cycle is that capsule, that sporangium, is going to be a place where meiosis takes place, and we're going to create haploid spores. But at this point in the life cycle, and you've probably seen this walking out in nature, looking at a cushy piece of moss growing under a tree and see these little thin wiry things coming off of it, and what you're looking at are both generations in the life cycle of a moss. So at the bottom right, you see a classic photograph of a bit of moss turf with some sporophytes growing off of it. Gametophytes in green, and sporophytes being the wiry structures above them. Next slide, true to form, meiosis occurs in that capsule. Remember, the capsule is also called a sporangium, or a spore container. And in this container, haploid spores are made through a process of meiosis. Meiosis is a process by which we start with a diploid cell and result with haploid products. And you'll remember It's an unavoidable part of his sexual life cycle. I might mention at this point, it's a truism concerning all sexual life cycles that meiosis is necessary because what it does is it counters the effects of syngamy. Syngamy tends to increase chromosome number, but meiosis reduces chromosome number. And so, if you're going to maintain chromosome number in a sexual life cycle, and it doesn't matter whether it's a sexual life cycle of an animal or a plant or any other organism, if you're going to maintain chromosome number, you need to counterbalance the effects of syngamy with this process known as meiosis. All right, so back to our moss plant. The very top of those wiry structures, I find the capsule, and I see a microscopic view of the capsule bottom far right. It's got a little lid on it, and that little lid is called an operculum. That's a term you don't need to know, but inside the capsule, you'll see clusters of spores. And those spores will ultimately be released, and when they are released, they will grow into more little haploid moss plants. They will grow into the green photosynthetic moss plants that we associate as gametophytes, because a haploid spore will grow into a haploid individual, a gametophyte. Well, that wasn't so bad. So, for your viewing pleasure, I have posted here a very generalized view of a moss life cycle. It's a moss life cycle diagram that was not terribly well labeled, so I threw a bunch of labels on it just so that it would actually fit what we've discussed on the last series of slides. So let's start off with the spores. And the spores are released from the capsule. And remember, the spores are made through a process of meiosis. They're haploid. They grow into little haploid moss plants. And haploid moss plants can be male moss plants or they can be female moss plants. They are gametophytes. Now, the male moss plant forms antheridia at the top of the moss plant, or gametangia, sperm containers. And those sperm containers contain sperm. The sperm are haploid, and since they're haploid and the gametophyte is haploid, those sperm have been made through a process of mitosis. Now shift your attention to the female gametophyte immediately below the male, and that female gametophyte will make an archegonium, or maybe many of them, archegonia at the top of the plant. But every single one of those archegonia will contain an egg cell in its base. And once again, the egg cell's haploid. And if the female gametophyte is haploid, the egg cell has been made by mitosis. Now, since these mosses live in a moist environment, there's going to be a film of moisture over the moss. At some point, the male is going to release its sperm cells from the antheridium, and the sperm cells will swim from the male to the female. Getting into the archegonium, a sperm will fertilize the egg cell in the base of the archegonium, fertilization, i.e. syngamy, and that fertilized egg cell will develop into an embryo. Now, the fertilized egg cell is officially called a zygote. The zygote will grow into a diploid embryo, a baby sporophyte, and the sporophyte develops right there at the top of the female plant into a long, tall, wiry thing consisting of a seta and a capsule on top. So sporophytes on the females, and those sporophytes, like I said, are the place where in the sporangium, in the capsule, an event of meiosis will take place and will make spores. And we're right back to the beginning. So, bird's eye view, I see a cycling between a gametophyte and a sporophyte. I see syngamy, or fertilization. I see meiosis. Now, the other thing I want to make sure you're aware of, and it's something I don't think I documented with any degree of emphasis before, but this is important, and that is that the dominant and conspicuous generation in a moss life cycle, dominant means you see it all the time, and conspicuous, you see it, it's obvious. So the dominant and conspicuous generation in the moss life cycle is the gametophyte generation. The sporophyte, on the other hand, is transient, it doesn't last all the time, and it's fairly inconspicuous. So the dominant generation in a moss life cycle, make sure you write that down, put a couple stars and exclamation points. The dominant generation in a moss life cycle is gametophyte generation. We're going to note that there's a big difference when we start talking about the ferns, and a big difference when we start talking about the pines, and a big difference when we start talking about the flowering plants. And those are coming up soon. So let's talk about ferns. Before we even get to the content on this slide, I'm going to remind you ferns are vascular plants. We could call them tracheophytes. They have xylem, and they have phloem, and they have cuticle. Would have to say that ferns are probably a little bit better adapted to life on land than mosses. They're still considered to be primitive plants. They're not quite as primitive mosses, but they're still considered to be primitive plants. Let's talk about the life cycle. The ferns that we see in nature are diploid. They're sporophytes. Big contrast to mosses. Remember I said that in mosses, the dominant generation, the dominant conspicuous generation of moss is gametophyte. In ferns, the dominant conspicuous generation is the sporophyte. When you see a fern and say, oh, gee, that is a fern, you are looking at a sporophyte. So our ferns make leaves, and those leaves are called fronds. And those fronds will make reproductive structures called sori. They're aggregations of sporangium found on the bottom surfaces of the leaves. You take a look at our photograph of the lower surface of a leaf and you see little dark dots. Each of those little dark dots is a sorus, or sori plural. And each sorus is an aggregation of sporangia or spore containers. If you take a look at the microscope picture to the right, you will see a bunch of little sac-like structures which are sporangia, and that entire structure you see there is a sorus. A sorus is an aggregation of sporangia, and each sporangium contains spores. And those spores are made by meiosis. Makes sense. The fern is diploid. The spores are haploid. They have to be made by meiosis. So our next job is to figure out what happens with those spores. So these haploid spores are going to be released from the sori on the underneath sides of the fern fronds. The spores will drop on the ground, and they will grow into tiny haploid gametophytes. That's that little heart-shaped individual that you see in the photograph. It's actually taken with a microscope. They're that small. They may only be a fraction of a millimeter across, but they represent an entire new generation in the fern life cycle. The sporophyte is the big, dominant, conspicuous plant, but the spores it makes grow into these tiny, small, relatively insignificant gametophytes that play a big role in the sexual life cycle of the fern. The fern gametophytes are non-vascular, unlike the sporophytes, but they are photosynthetic. They also have the capacity to make both antheridia and archegonia. Now, you can make both of them on the same individual. So there I see antheridia, central photograph showing you a small container, the bottom of this gametophyte, and it contains small red sperm cells. On the other hand, the archegonia look vaguely like the archegonia that we found in the mosses. They're nowhere near as long, and they seem to be positioned upside down, but they are found embedded in this moss gametophyte, and I see what appears to be a chimney-like structure, and at the base of that chimney-like structure there is going to be an egg cell, comparable to what we see in the mosses. So we're setting up a situation where we are going to have a fertilization event. Now, remember, in mosses, water was our agent of fertilization. We had to have a layer of moisture in order to get the sperm from the males to the female. The same is going to be true for our gametophyte here. The gametophyte lives right smack dab on the ground in a relatively moist environment, and there can be a complete layer of moisture on that gametophyte, and there can be the release of those sperm cells from the antheridium, and those sperm can in fact swim from the antheridium to the archegonium and therefore fertilize that egg. We're going to see that on our next slide. When gametophytes are covered with a layer of moisture, sperm are released from the antheridia. The sperm are shown in the illustration lower left, and they look a little bit like the sperm that I might find in a moss plant, but they do the same basic thing. They swim through this film of moisture, they enter the archegonium, and they fertilize the egg cell at the base of the archegonium. The egg cell is fertilized and it becomes a zygote, and it grows into an embryo, which is a tiny baby sporophyte, which grows into a smaller version of an adult fern plant. That sporophyte you see at the far right, it's a tiny version of the adult sporophyte plant that you and I see when you go walking through the woods. It's a tiny diploid individual that has just started out growing from a fertilized egg cell that has remained in an archegonium where a fertilization event has taken place. Now, in short order, at some point, the gametophyte is going to die. And the sporophyte is going to be able to maintain itself as a free-living individual once it forms roots and once it starts doing photosynthesis, and it will be able to persist for year after year after year after year. Well, I guess I gave it away on the previous slide. The tiny diploid sporophyte grows from the female gametophyte, but soon develops its own leaves and is able to photosynthesize on its own. And at some point later in its life, this diploid sporophyte will make sori and haploid spores from meiosis, just like it did in the previous generation. And we're right back to where we started again. So, Dr. Kness, looks like you've got another life cycle summary for us. And there's plenty of these available online, folks. If you don't like this life cycle, or you don't like the bryophyte life cycle I showed previously, or if you don't like any other of these diagrams you've been seeing in this presentation, Google this stuff. There are plenty of artists' illustrations that might tickle your fancy more than the ones I've chosen here. But let's go ahead and take a look at the fern life cycle summary and reconstruct what we already know. The fern as we know it, the dominant conspicuous generation, is a sporophyte. I see it down there at 6 o'clock. That fern produces fronds. The frond can produce sori on the underneath sides of the fronds, and those sori are aggregations of spore containers or sporangia. And a sporangium is where meiosis takes place. Haploid spores are produced, haploid spores are released, haploid spores grow into the young gametophyte, which is a tiny, non-vascular, photosynthetic, heart-shaped individual that lives right down there on the ground underneath the sporophyte in an area that is undoubtedly moist, and a complete layer of moisture will afford us the ability to have a fertilization event. On that gametophyte, I'm going to have antheridia, and an antheridium is a sperm container, and it contains haploid sperm. The haploid sperm are made by mitosis. The archegonium is the female gametangium. It's an egg container. It contains a single egg, also produced by mitosis. A fertilization event occurs after the sperm have been released from the antheridium and swim to the archegonium, fertilizing the egg while it is in the archegonium. A zygote results, and the zygote, brand spanking new diploid cell, grows into an embryo, a baby sporophyte. And that baby sporophyte at some point becomes an independent, free-living, photosynthetic plant. But before it does, it starts off as being initially dependent upon the female. So my illustration there shows me the zygote transitioning to become the young sporophyte on the gametophyte that will in turn grow to become a mature individual. We're about to talk about seed plants, but I want to make one point clear. And I think it's one I've made already, but it's when we're taking a look at the mosses and the ferns, which we've just covered in terms of the life cycle, please remember the mosses are more primitive. The ferns are a bit more advanced. The ferns qualify as tracheophytes because they have xylem, they have phloem, and they have a cuticle. Those are all adaptations to a terrestrial existence. But I'll tell you what, both the ferns and the mosses are still, I guess maybe we could say they're still dependent somewhat on water. If we think that the plants evolved out of algae like organisms that were living in an aquatic environment, obviously, if you have xylem and phloem and a cuticle, you're taking a step out of that environment. But remember, when we were taking a look at the life cycle of the mosses and the life cycle of the ferns, in both cases, water was a requirement for fertilization. That harkens back to the evolutionary history of these plants. Yes, they live life on land, but at one point in their life cycle, and that's fertilization, they are beholden to water as a medium to get that sperm from the antheridium to the egg in the archegonium. Now, in a minute, we'll be talking about seed plants, and seed plants have avoided the water requirement. We will see that seed plants have xylem. We will see that seed plants have phloem. We will see that seed plants have a cuticle. Seed plants are tracheophytes. But one other additional benefit that the seed plants have, at least in terms of life on land, is they are no longer dependent upon water as a medium to get a sperm from a male to a female. Instead, we have a pollen ovule arrangement. And we haven't seen pollen and ovules yet, but we are going to talk about them in the context of the seed plant life cycle. So let's start with the life cycle of the pine. Pines are members of a group of plants called gymnosperms. And the word gymnosperm, it's spelled Gymnosperm. The word gymnosperm means naked seed. So these plants make seeds that are not enclosed and not protected in a structure like an ovary that we would find in a flower. So let's talk about the pine life cycle. The pine trees we see in nature are diploid. They are sporophytes. The sporophyte generation in the pine life cycle is the dominant and conspicuous generation. Now, these sporophytes, you and I both know, make cones. And the cones that we find on a pine tree come in two different flavors. There are male cones and female cones. However, I need you to know the official term for a cone is the word strobilus. So I've got a male strobilus, I've got a female strobilus, or in plural, I could talk about male strobili and female strobili. In any event, be it a strobilus, be it a cone, that structure is the reproductive structure found on the pine tree. It's kind of comparable to the flowers that we see on flowering plants. Now, the male strobili are only found on the pine tree a certain amount of time during the year. They're not permanent structures. They are generated during late spring to shed pollen during early summer. So if we take a look at our pictures there, you will see a pine tree. You will see a cluster of male pine cones. They're kind of yellowish in color. You will see a longitudinal section through a male pine cone, and you'll see it contains little itty-bitty sacs, and each one of those itty-bitty little sacs contains a whole bunch of haploid pollen grains. Now, haploid pollen grains are tiny haploid male individuals. They constitute the gametophyte generation. And I know that's not on the slide, but I need you to note that the pollen there in the male pine cone, in the male strobilus, those are tiny haploid male individuals. those are male gametophytes. And note, it says there that pollen are made by meiosis. Well, let's talk about ovules now, because ovules are made by meiosis as well. Starting over on the left, I see my pine tree. I see a couple of female pine cones or female strobili. I see a cross section through a female strobilus and a close up view of what appears to be the scales on the female strobilus. And if you take a look at those scales carefully, you'll see kind of an oval shaped structure embedded in each scale. That's an ovule. So that's a tiny haploid female. And an ovule is a gametophyte as well. So here I've got a big, giant diploid plant featuring male strobili, featuring female strobili, and on the male strobili, I've got tiny male gametophytes, and on the female strobili, I've got tiny female gametophytes. My goal is to get the males from the male strobili to the female strobili. Now, that process of moving a pollen grain from a male to a female is called pollination, and it is necessary to get us an event of fertilization. It's something we talked about not too long ago in a different presentation, but we will go into it again in the next couple of slides. All right, we already made the observation that in late spring, early summer, pines shed pollen. And you're aware of the yellow dust that gets all over your car and all over windowsills, and it lies on puddles in the street, and it's just everywhere. You and I are not allergic to pine pollen. I promise you, it's much too big for us to be allergic to, but it is a hassle. So let's take a look at a bullet point. The haploid pollen are going to be shed from the male strobili and are going to be carried by the wind to the female strobili. That process is called pollination. More officially, the term for wind pollination is anemophilous pollination. That's ANEMOPHILOUS. That's a mouthful. Anemophilous pollination. So, a male pollen grain in a pine tree is really small, but it's still a male and it's still capable of making sperm. So I see my picture on the far left, some male strobili releasing pollen. That's the cloud. I see a whole bunch of pollen grains, and I see a close-up view of a pollen grain, and each of the pollen grain looks almost like a little Mickey Mouse head. And the pollen grain itself is the central cell, and on two sides we got a couple of wing-like structures that will aid in that anemophilous or wind-motivated pollination process. Now, if you take a look at the next picture, you will see a couple of red-stained pollen grains, and you'll see each one making a tube. So once the pollen lands near the ovule, it's going to make a tube, but focus on the little itty-bitty dark-stained structure traveling down through that tube. That is ultimately going to be a sperm cell. So I've got ovules on the female strobili, and those ovules contain eggs. The pollen is going to land near the ovule and form a pollen tube that will grow in towards the egg cell. So if I take a look at the ovule illustrated at the top of the slide, on the right-hand side, I see a couple little dark structures. Those are little egg cells sitting in the ovule. And I look a little bit below that, and I see an artist's rendition of a pollen grain growing its tube in towards the egg cell ovule, which is basically illustrated there as B. So what we got is setting up a fertilization event. And in this case, I have no water requirement because the pollen has been direct delivered to the female through an event of pollination, and the growth of the pollen tube delivers that sperm directly to the female. So once again, no water required. A really, really nice adaptation to life on land. Something that the ferns don't have and something that the mosses don't have. All right, close-up view from our previous slide. The pollen tube carries the sperm to the ovule and the egg is fertilized. Fertilization is syngamy. So remember, pollination is distinct from fertilization. Pollination is the agency of getting the pollen from the male to the female. And remember, those pollen are tiny male gametophytes. And then fertilization is the actual fusion of the sperm with the egg cell, which is the goal ultimately of pollination. And the fertilized ovules remain on this female strobilus, and they will ultimately develop into seeds. So there's a close-up of view of an ovule after fertilization, and you'll see it's got an embryo that's a baby pine tree labeled E. I see there's a food source surrounding that embryo labeled FS. And then there's the seed coat, the hard outer covering that's going to protect the seed between now and whenever that seed actually germinates. And in pine trees, as a rule, it's about 2 years. So here I've got a brand spanking new individual containing its own little time traveling capsule, which is this thing called a seed. And a seed is developed as a consequence of the fertilization of ovules on the female strobilus. So let's wrap it up here. The seeds develop on the female strobilus, and the female strobilus, after a while, it's going to dry out, and the scales on the female strobilus are going to open up, and the seeds are going to be shed. Typical female pine cone, there on the lower left, having opened, and I see a bunch of seeds lying next to it. And those seeds ultimately will germinate and grow into a brand new pine sporophyte. Now, once again, looking at that pine tree, in my heart of hearts, I know it's a sporophyte. It's diploid. It's got adaptations to life on land. It's a tracheophyte. It has xylem, it has phloem, has a cuticle. And not only that, but we also know that it no longer depends on water in order to get the sperm from the male to the female. The male's tiny individual gametophytes are shed from male pollen grains. They are carried by the wind to the female. They're direct delivered to the female. They form a pollen tube that grows into where the egg is. So when all said and done, the fertilization event takes place without the agency of water. Another really nice adaptation to life on land. Hold that thought. That's going to be a theme that will be repeated when we take a look at the flowering plant life cycle. Next, we'll be taking a look at the lily. All right, well, here's our last plant life cycle we need to be concerned with. Breathe a sigh of relief, I guess. You might be a bit overwhelmed with what seems to be a whole bunch of complicated life cycle stuff, but once again, they all bear similarities to each other. They all follow a basic theme, and that involves an alternation of generations life cycle, an alternation of something that's haploid, gametophyte, with something that's diploid, a sporophyte. And meiosis and syngamy are the transition points between those two generations. Now, once again, we've identified some very, very nice adaptations to life on land, and those adaptations involve the traditional tracheophyte adaptations of xylem and phloem and cuticle, but also with the institution of pollen and ovules, we avoid the water requirement for fertilization. See, way back when, mosses required water for fertilization, ferns required water for fertilization, but pines don't. And neither, turns out, are our lilies or our flowering plants. So the lily plants we see in nature, as our example, are diploid, just like the pine trees. They're sporophytes. They are diploid organisms. They are produced as a consequence of a reproductive event. Well, these flowers, they grow, they do photosynthesis, and they make reproductive structures that you and I call flowers. And a flower is nothing more than a modified shoot. And all the parts of a flower are technically modified leaves. So I take a look at my lily flower down below, and I know it's got all those nifty parts. It's got sepals, it's got petals, it's got stamens, and it's got pistil. And the stamens and the pistil are the reproductive parts of the flower. So let's take a look at the stamens. Now, I know a stamen is a long filamentous structure that has something called an anther at the top. And the anther of the stamens contain haploid pollen grains, or tiny male gametophytes. And those male gametophytes are ultimately made by meiosis because this is a diploid plant and it's creating haploid pollen. Hold that thought. Now, take a look at the picture of the lily, and I see a close-up view of those red anthers, and a cross-sectional view of those anthers show me little chambers that are officially called locules, Locules, and those locules contain the pollen. And at some point, those locules are going to release the pollen, and the pollen are going to be transferred via air, via wind, via bee, butterfly, what have you, is going to carry that pollen from the male to the female. Now, take a look at the same picture and follow the pictures at the bottom of the slide. I see a close-up view of the pistil. I see isolated there, and I see part of the style, and I see the swollen lower portion of the pistil that we call the ovary. And I notice in cross-sectional view that ovary contains ovules. I see 3 locules. Remember, locules are chambers. Same term we use in the anther, and each of those locules appears to contain a couple of ovules. So the ovary of the pistil contains haploid ovules, which are tiny haploid females, or we should specifically say female gametophytes. And they're made by meiosis too, because once again, the plant is diploid, and in order to get from a diploid plant a sporophyte to a haploid gametophyte, be it a pollen grain or be it an ovule, meiosis has to be the operation that does that. All right, so what happens next? The haploid pollen are shed from the anthers and are going to be carried to the stigma of the pistil. That event is called pollination. And that event can be moderated by insects or by bats or butterflies or the wind. In some cases, you got plants are actually pollinated through the action of water. But regardless, I'm carrying the tiny male gametophyte to the tiny female gametophyte. The pollen is a tiny haploid male, and the ovule is a tiny haploid female. Each pollen grain, despite the fact it's really small, it's only 2 cells big, believe it or not, has the capacity to make sperm. If you take a look at the illustration bottom right, you will see a pollen grain. You'll see it forming a pollen tube, and you'll see 2 sperm nuclei traveling down that pollen tube. That's the male doing its thing. So each ovule in the ovary of the pistil contains an egg. It's typical, like we've seen before. The pollen grain lands on that receptive surface of the pistil called the stigma, and it forms a pollen tube that grows downwards through the style towards an ovule in the ovary. Now, you think about it, there's probably a whole truckload of pollen grains on any one stigma, but there's a whole truckload of ovules down there in the ovary, and I need a whole truckload of pollen grains to form a truckload of pollen tubes to fertilize a truckload of ovules in the ovary. And as a consequence, a fertilized ovary can contain a significant number of seeds, each one generated from an event of fertilization. But now let's focus on what goes on with a single pollen grain. And once again, 2 sperm nuclei are carried through the pollen tube as it grows downwards through the style of the pistil towards the ovary. Okay, picking up from where we left off, the pollen tube carries 2 sperm to the ovule. Now, take a close-up view of the ovule. It's in the process of being fertilized. I see it down there on the lower right. And you'll see 2 arrows. There's 2 sperm that are going to be delivered to this ovule by 1 pollen grain. Sperm number 1 fertilizes the egg, and a zygote is formed. You'll see the arrow indicating that fact. That zygote will grow into an embryo, which is a baby plant, and will ultimately become a brand new lily plant once that seed germinates. But there's a second sperm, and the second sperm contributes to the formation of the food source for that developing embryo, and that food source is being developed right smack dab in the middle of the ovule there, and it's called endosperm. And eventually, the endosperm is going to be a starchy material that will serve as a food source for the embryo while this seed is waiting to germinate. Obviously, if this seed is set in springtime of 2023, it's not going to germinate until maybe springtime in 2024. That's a full year that that seed has to survive. That's a full year that that embryo has to survive. And so we've got this built in food source. Now, the process of two sperm resulting in two products, one being an embryo, one being the endosperm, is called double fertilization. We'll probably invoke that phrase later on when we start talking about angiosperms or flowering plants in more detail. Okay, but in lilies, make sure that you understand the process is called double fertilization. It's descriptive. It tells me I got two sperm, I got two events of fertilization taking place. Now, one of those events is actual fertilization, actual syngamy, the fusion of a sperm cell with an egg cell, creating a diploid zygote. But the other is kind of like a form of fertilization, in which case I'm creating this brand spanking new tissue that's going to serve as a food source for that developing embryo. So once that fertilization event happens, those fertilized ovules remain in the ovary of the pistil, and they're going to ultimately develop into seeds. We understand that a fertilized ovary is a fruit. Doesn't matter whether it's a watermelon or a tomato or an apple, but inside of that fruit, I'm going to find these fertilized ovules, which are going to be seeds, which once they're released from the fruit, can grow into a brand spanking new apple tree or watermelon vine or tomato plant. And each seed has the standard set of components. There's an embryo, brand spanking new diploid sporophyte plant. There's a food source. In this case, we're going to call it endosperm. And then it's all surrounded by a seed coat. See this on our next slide. All right, as time goes by, the parts of the flower are going to shrivel up and die, and we're left with this fruit. A fruit is a post-fertilization ovary. So on bottom left, you see a couple dried capsules, which are the remains of the ovaries from a lily. And follow the arrow, you see those capsules contain seeds that are going to be shed. And each of those seeds contains the standard seed components. I see a little bit of a cross-sectional view of a seed immediately above, and the things you want to be aware of are the seed coat surrounding the whole thing, the endosperm, which is the food source for the developing embryo, and the embryo itself is kind of tucked towards the bottom of that particular illustration. That's what ultimately will go into a brand new lily plant. That was a lot of content. We've basically got command of 4 different life cycles characteristic of some rather unique groups of plants. We've got a moss life cycle under our belt. We have a fern life cycle under our belt. We got the pine life cycle under our belt. We have the lily life cycle under our belt. And we've acknowledged the fact there are some evolutionary modifications, or let's say adaptations, that have basically made life much more successful for plants on land, which, to be honest, is a pretty inhospitable environment. So we'll talk about these modifications. Now, on the right-hand side of the slide, I see kind of an evolutionary progression. Our ancestral plant was an aquatic plant. It was probably a green algae. It was living in the water. And at some point, we made a transition to life on land. And so the most primitive plants that demonstrate mostly aquatic like characteristics are mosses. Mosses are not especially well adapted to life on land. As a consequence, you find them in very limited circumstances. And they're limited because they have no cuticle, they're limited because they have no xylem, they have no phloem, and they still have a requirement for water as a medium to get sperm from the male to the female. Mosses are limited in terms of what they can do in a terrestrial environment. Ferns are a little bit more advanced. They're a little bit better adapted to life on land. They have a cuticle, they have xylem, they have phloem, but as far as the water requirement, they still got it. They still depend on water to get that sperm from the male to the female. Ferns have made a big step onto the land, but you know what? They've got one big toe still in the water, which is that fertilization issue. Then we move to seed plants, who've made the complete break from life in the water because they've got it all. And that would not just be the pine, but it will also be the flowering plants. They've got xylem, they've got phloem, they've got a cuticle, and they've completely avoided the water requirement for fertilization because they've got pollen and they've got ovules. And we do direct delivery of the pollen to the ovule, forming a pollen tube, carries the sperm directly to the female, no water required. Makes life much more successful in a terrestrial environment. So let's summarize these advantages we've mentioned. And I need you to make sure that you can list those. These are important. Advantage number one, cuticle. We understand that it's a layer of wax over the surface of the plant. It prevents desiccation in an otherwise inhospitable terrestrial environment. Advantage number two, we've mentioned stomates and guard cells. We haven't specifically pointed a finger at anything, but land plants, as a rule, have stomates, and guard cells regulate those stomates. And the stomates provide a pathway for gas to move into and out of the plant. But you know what? It's also an avenue for potential water vapor loss. And life on land means that if we've got some kind of opening to deliver gases, we'd probably want to regulate it so that when evaporative pressure is extreme, all we have to do is close those stomates. And yes, we restrict our gas exchange, but the most important thing is we restrict the amount of water vapor we're losing. Let's take a look at our next slide and continue on with this list. Advantage number three, xylem and phloem allows physical separation of roots for nutrient and water acquisition and leaves for light acquisition. The fact of the matter is, if you are a plant living in an aquatic environment, light is pretty much almost all around you because it's diffracted as it enters the water, but you're also pretty much surrounded by all the water and all the nutrients you need. You really don't need structures specialized for collecting water and minerals, and you don't need structures specialized for collecting light. That becomes a big issue when you take yourself out of the water and you put yourself on land, because the light that you need and the water and the minerals that you need are spatially separated. They're physically separated from one another. The light's above the ground and the water and the nutrients are below the ground. So therefore you need a shoot system, you need leaves, and you need roots. And the xylem and the phloem makes it possible for you to connect those two. And life would not be possible if you didn't have that connection. And the other thing is, it also makes it possible for you to build a much larger plant body. It's one of the reasons why trees are so darn big, because you've got this vascular tissue that allows you to connect leaves way, 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 way up above the ground with roots that are way, 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 way down below the ground. It's one of the reasons why mosses are so small, because they don't have that vascular tissue that affords that ability to make those connections. Advantage number four has to do with the diploid generation being dominant. Remember, that's the sporophyte. And the diploid generation is dominant in the ferns, and the sporophyte is dominant in the pines, and the sporophyte is dominant in the flowering plants. Now, in the diploid generation, the thing we've got is genes are paired because chromosomes are paired. And if genes exist in paired combinations, those unique combinations, those alleles is what they're called, increases the potential for genetic variability. There's a lot more potential for genetic variability in a diploid individual versus a haploid individual. So that variability and ultimately flexibility for life on land exists in the sporophyte, and that's what makes, at least in part, the seed plants, the pines, the flowering plants, and the ferns a lot more, let's say, successful and more evolutionarily flexible than the mosses. Advantage number 5, we mentioned pollen and ovules, which are unique to the pines and unique to the flowering plants. And we harped about this before. Because we've got pollen, because we've got ovules, there's no dependence on water for fertilization. We saw a dependence for water for fertilization in the mosses. We saw it in the ferns, but we don't see it in the pines, and we don't see it in the flowering plants. The male gametophyte, the pollen, is direct delivered to the female. No water required. And then finally, advantage number 6, at least as far as the seed plants goes, our seed has a structure and has a feature of dormancy that allows it to succeed in seasonal habitats. I can make myself a seed and I can let it sit there for a full year, maybe 2, maybe 3, maybe 4, but as long as that embryo is dormant and as long as that embryo has a food source, and as long as that embryo and that food source are protected by a seed coat, you've got an entity that is resistant to the hostilities of a terrestrial environment. Okay, so for our last slide, wrapping it all up, I've got that same progression that I had on the first slide. I see my ancestral aquatic plant, I see my mosses, I see my ferns, I see the seed plants, pines, I see the seed plants, flowering plants, and I notice I've got some numbers next to those groups indicating those adaptations that I just listed on the previous 2 slides. So ultimately, life on land is tough. And if you are going to evolve out of an aquatic environment like you're an algae and you want to make good in life on land, you need to have some adaptations. And those adaptations will improve your fitness. And remember, fitness is your ability to survive and your ability to reproduce. So adaptations, advantages number 1 through 6, make that possible. And so therefore they have demonstrated an evolutionary history of success in our plants, ultimately the most successful of those plants being our seed plants. And we're going to review this again once we start talking about mosses and ferns and and pines and flowering plants in more detail in maybe a future presentation. So hold that under your hat because we'll be visiting it again. And obviously, it's going to be of critical importance on your next exam. And this is a lot of content. I understand it's a lot of content. Please take your time. Please invest a lot of energy. Please be dedicated in making sure you know it frontwards, backwards, upwards, downwards, and sideways.